All right, so during our first lecture, I gave you guys a very thorough background knowledge of what you should know before understanding metabolism. Remember that it is our goal to then be able to describe all of the reactions for central metabolism. For example, starting this week, we're going to be covering the reactions and mechanisms for glycolysis. Okay, and obviously, glycolysis, you should be able to decipher from the word glyco, meaning sugar, lysis, breakdown. Okay, so it's the reactions that you use to break down sugars, okay, and in particular glucose. So let's take a step back and sort of think about why we're interested in understanding glycolysis. That is because obviously you can appreciate that the food that you eat, for example, if you get up in the morning and you eat some bread, or if you go and eat potato chips and things like that, okay? A lot of these food products that you eat every day contains a lot of starch. For example, your rice or your noodles. Starch, again, remember, it's composed of mainly amylose and amylopectin. If you guys remember from Biochem 1, amylose is a polymer of glucose, okay, linked via alpha-1,4 linkage. Okay, so that's amylose. It's a sort of linear polymer of glucose. If you have amylopectin, starch actually has quite a bit of amylopectin inside. What it is essentially your linear polymer, your linear alpha-1,4, but then you also have alpha-1,6 bonding, okay? But regardless, amylose, amylopectin, these are all polymers of glucose, okay? Or let's say you go out and get a boba tea somewhere, and obviously boba tea, they will have sugar, okay? Sugar, one of the most commonly found sugar that you use as a consumer product will be sucrose, okay? And remember, sucrose is a disaccharide of glucose and fructose, and it's usually extracted from sugar beets or sugar cane, okay, like that. Taiwan used to produce a lot of sugar canes. All right, so you can appreciate that if you look at these two common food that we eat, and you will notice how they are polymers or dimers of sugars, and of which they contain glucose. Okay, for example, here's all glucose, and here is one glucose plus one fructose. So you'll understand that glucose is a large percentage of your daily diet. Okay. In addition, okay, through evolution, biology have evolved ways of providing structures, things like that. And for example, if you look at plant biomass, right, plant biomass is composed of cellulose, hemicellulose, and lignin, okay? And cellulose, recall that it's also a polymer of glucose. However, it's beta-1,4 linkage, okay? That's what cellulose is. Hemicellulose hemicelluloses, they're copolymers of various sugars, and these sugars could be glucose, could be xylose, could be arabinose, glucuronic acid, whatever. Okay, so which is why it's called hemicellulose. It's not exactly polymers of glucose. It's copolymer of various sugars. Okay, and inside of plant cell wall or plant biomass, so if you take a cross section of the plant cell wall, and it's going to look something like this. Okay, you have these cellulose or hemicellulose fibers. Okay, so each strand corresponds to a strand of sugar polymer, and inside you see the difference between the white and the black. Okay, the white ones represent cellulose, and between the white ones sometimes you have these hemicelluloses, right? So that's just, that's just how plants are able to provide a sort of structure to its cell wall. And most importantly, the structure is actually held by this thing called lignin. Okay, so lignin is a polymer of aromatic alcohols, right? So if you were to look at this structure right here, immediately you see that there's a lot of sort of aromatic residues like benzene rings, that kind of stuff, and they're going to be linked together, right? And when they're linked together, they sort of form the support for the cellulose fibers, okay? So, have you ever thought about why if plants, like plant biomass, okay, if they are polymers of glucose, how come they're not so easily degraded, okay? Think about that, right? Obviously, your cellulose beta-1,4 linkage is very tough. Yes, it's tougher than amylose, okay? But yeah, you know, there are microorganisms that can degrade cellulose readily easy, but yet your plant leaves or your plant trunks, they're still there, right? They're still there. And that's actually, it turns out it's because of the lignin, okay? Because of the lignin. Lignin is not very easy to degrade, okay? For obvious reasons being, you know, it's aromatic. So you guys remember from organic chemistry, if you were to do chemistry to like benzene, it's not going to be very easy, right? Because benzene is super stable, right? It's very stable. So if you want to do any reaction on it, it's not a very easy thing to do, okay? So lignin is actually one of the bottleneck for, let's say, my particular research, like renewable energy or chemical production. Okay, so people that are interested in using waste plant biomass and using that to produce biochemical, for example, one of the biggest challenge is how to get rid of the lignin, okay, because lignin is very tough. All right, but whatever. Our focus is on once you were able to degrade plant biomass and release cellulose, okay, and then you have these enzymes called cellulases or hemicellulases. If there's an enzyme that is reactive in particular to xylose, it's going to be called xylanase, okay? Now, your cellulases, there are various kinds, there are, you know, different types of cellulases, but primarily you can break them down into endo and exo, okay, just like your endonuclease and your exonuclease, okay? And endocellulase will randomly cleave your cellulose fiber, and it's going to break it down into smaller pieces, okay? And then you can have these exocellulases that will chew them from the outside in, okay? And that will degrade them into two, like a dimer or a tetramer of sugars, okay? So they will go from the outside. And then subsequently, these short oligosaccharides, these dimers or tetramers, can then be further broken down into monomeric sugar. Like for example, if you have a dimer of glucose beta-1,4, this is called cellobiose. You use a thing called beta-glucosidase, okay, glucosidase, because this is a glycoside bond. So glucosidase, okay, glucosidase, okay, so it degrades the glycoside bond, or the glycosidic bond, then you'll be able to liberate the two glucose. Okay, so this is how we go from something that is complex, okay, something that is a polymer of glucose, and all the way down into the single monomers of glucose. Okay, so this is how we break it down. All right, so the key topic that we're going to be talking about will be glycolysis. Okay, so now that we have glucose, and our question then is, how does glucose get used? Okay. This reaction is called glycolysis, and it converts glucose down to pyruvate. Okay, remember I say pyruvate is like the center of metabolism, because once you have pyruvate, you can pretty much make everything else. Okay, this glycolysis pathway, it's also called the EMP pathway, named after three scientists who discovered the glycolysis pathway. Right, and just for your information, okay, there are more than one type of glycolysis, okay? But primarily we're going to only focus on the EMP pathway, and that is because this is the pathway that human cells use. Okay? But in bacteria or in a lot of other archaea organisms, they have other types of glycolysis. They look similar, but they have a little bit of differences here and there. Okay? And glycolysis can be split into two phases, like we talked about last time. The first phase can be called preparatory phase or activation phase, or what I like to call it like an investment phase. And then the second phase is a payoff phase. Okay? The investment phase, essentially, what it does is you have to put energy in the form of ATP, okay, onto glucose, such that you can activate glucose and do reaction on it, okay? Otherwise, glucose is pretty stable. So you spend 2 ATP to convert 1 glucose, 1 6-carbon molecule into 3 carbon molecules, and 2 of them, okay? So 2 3-carbon molecules. And these 2 3-carbon molecules are whatever phosphates, okay? So they're high energy. And now you have these 2 3-carbon molecules, then they can do your payoff phase, okay? And for each 3-carbon molecule, you can generate 1 NADH, 2 ATP, okay? And since you have 2 of these, okay, so you can generate a total of 2 NADH and 4 ATP, okay? You can generate a total of 4 ATP. So if you subtract the 2 that you invest, the net reaction of glycolysis will be 1 glucose to 2 pyruvate plus 2 ATP plus 2 NADH. All right, so this is the sort of, what do you call it, the net reaction for EMP pathway, okay? All right, and we're going to be talking about all of the reactions, each reaction in detail, and we'll talk about their mechanism, right? The first reaction is called hexokinase. Why do you think it's called hexokinase? Kinase is obvious, right? Kinase is an enzyme that will use ATP as a phosphate donor to phosphorylate something. Okay, so blah blah kinase is an enzyme that phosphorylates blah blah using ATP. Okay, kinase. Hexo, where do I get hexo from? Hexose. Okay, it's a 6-carbon sugar. Hexose. So it's like hexose kinase. People got lazy, so we call it hexokinase. Okay, so hexokinase. So it's gonna sort of turn glucose into glucose 6-phosphate, okay, using ATP. All right, so let's first talk about transport of glucose. All right, glucose looks like this. All right, this guy is glucose. And remember, shorthand will be GLC for glucose, okay? And the first thing that glucose will have to do is somehow get through your cell membrane. So this guy is cell membrane. Okay? Somehow it's going to get through the cell membrane. Okay? First question for the audience. Do I need a transporter? Yes? Okay? All right. I'll believe you. So you need to have a transporter. Okay, now. Does the transporter require ATP? It does. Does it? You sure? Okay. All right. So one of the mechanism here, you can use ATP. Okay. This is called a what kind of transport? Active transport. Okay. This is called active transport. Okay? Can glucose come in without using active transport? Maybe? Maybe okay? Okay, yes, it can. Okay, remember we talked about 3 types of facilitated transport, right? And one of it was this thing called a permease. Okay, so glucose can also come in using a glucose permease. Okay, now remember, okay, glucose permease does not require ATP, but the active transport does. Okay, you should be able to differentiate the two, what the two is for. Okay, permease, it's reversible. Okay, so it's like it can go in, it can go out. Okay, depending on the concentration. Okay, so permease responds to concentration. Okay, whichever side is more concentrated, it's going to go the other side. Okay, now active transport, remember, is a way to sort of pump in stuff. Okay, so for example, if inside the cell the concentration for glucose is already high, but you want to make it higher, okay, then you will have to use the energy of ATP to overcome the chemical potential. Make sense? 可以啦，有没有听不太懂 ？OK 好 ，Alright. So glucose comes in. Alright, so you'd have glucose coming in. Glucose. Okay. So then, reaction 1. Hexokinase. Okay, shown over there. All right. So hexokinase, what it's going to do, okay, is that it's going to use ATP and then put a phosphate onto this guy. Okay, so I'm going to draw ATP right here. Here I'm just going to call AMP because AMP doesn't matter. O. Okay, so AMP, this is the second phosphate, so this is called ADP, including the third phosphate is ATP. Okay, so my shorthand for ATP. All right, remember I said any enzyme that uses ATP will probably have what as the cofactor? What as the cofactor? It's on the board. I mean, it's on the slide. 来嘛，给点 response 好不好 Magnesium. Thank you. Magnesium. Okay, so it's going to have magnesium as a cofactor. So you have magnesium 2 plus. Okay, so it's sort of used to stabilize ATP. Okay, something that looks like that. All right. So I can tell you right now, this reaction, it's sort of kind of like a transesterification reaction. It's kind of like that. All right. So we're going to start with a nucleophile and an electrophile. Okay. In this case, nucleophile is going to be the sugar. Okay. The oxygen on carbon 6, okay, because we're making glucose 6-phosphate, okay, the oxygen, the hydroxyl group on carbon 6, the oxygen there has 2 lone pair electrons. Okay, so if you have lone pair electrons, it's a nucleophile. It's going to do a nucleophilic attack on a electrophile. All right, here, this guy, who is the electrophile? I mean, it's going to be glucose attacking ATP, right? I hope, okay, right? So if you look at the structure of ATP, or just look at a structure of phosphate right here, who do you think, or which atom do you think will be the electrophile? Phosphorus, okay? The P, right? Obviously. 好了 ，OK， 来哦。So the 2 electrons are going to like fly over here, attack the phosphorus. And what happens? Obviously, here you have a pi electron pair and it's going to pop up. We're okay? Okay, right? And what do you think you get? Okay, so I'm going to draw the rest somewhere here. Okay? And you're going to get, I'm going to call this entire thing just glucose. Okay, so you're going to get glucose. Here's the oxygen. Okay, so this is carbon 6. Carbon 6. Okay, here's the oxygen. Oxygen is going to be attached to a phosphate. Oh, no, yeah, it's going to be attached to a phosphate. And here you have the first O, second O, and third O, another O, P, whatever. So this is A, M, P. Okay? And the reason why enzymes can sort of mediate this formation of this oxyanion looking thing is because you have a magnesium here, okay? So then, it becomes like that. And actually, never mind. So pretty much that, all right? So you form a new bond between this oxygen atom to this phosphor atom. Okay, Ma? Okay, we're good. All right. And I was saying the reason why you can form this sort of charge, this charge here, is because you do have a stabilization provided by the magnesium. Okay, so that you can sort of promote the ability of these oxygens to withdraw sort of the electron density away from phosphorus, and it sort of stabilizes the intermediate. Okay. Now, what do you think happened next? Something is going to happen to this guy, this oxygen. It's kind of like an oxyanion, right? Kind of. And remember last lecture I said the only two things that oxyanions can do? What are the two things? What are the two sort of subsequent actions that oxyanions can do? The first thing I can do is grab a proton, right? Grab a proton from the solution and it becomes an OH. Okay. What's another thing that it can do? Jump back down, right? So you can take the charge and then pop it back down, okay? But when you pop it back down, now the phosphorus will have too many bonds. Okay, then what do? You gotta kick out an oxygen somewhere. You can kick out the glucose, but then that will just take you back to where you started. Or you can kick out the ADP. Okay, make sense? Right. So once you kick out the ADP, then you're left with glucose, O, and then phosphate. Okay, so this is G6P. Okay, and then the rest, this part is ADP. Okay, does that make sense to you guys? I mean, it's really straightforward. Okay, so this is the mechanism for kinases. Okay, so obviously, all other kinases behave this way. Okay, so you just covered like, I don't know, maybe 5% or 10% of mechanisms that found in central metabolism. Okay, there's a bunch of kinases around. All right, are we good? Any questions on how this happened? This is not magic. Okay, this is chemistry. All right. So once you form glucose 6-phosphate, okay, sort of this phosphate, not only does it have higher energy, but turns out it sort of serves as a mark. By mark, what do I mean is that this guy cannot leave the cell, okay, because the glucose transporter or the sugar transporters that you have for taking in glucose does not recognize, at least not well, does not recognize glucose 6-phosphate. Okay, so as a result, if you did the activation step, the first reaction here, you sort of signals this particular glucose, it's going to be used for something. Okay, so it's not going to leave your cell. Okay, so because it now has a phosphate, it does not leave the cell. Okay, turns out in a lot of bacteria, so if you were talking about E. coli or bacillus, okay, turns out in many bacteria, glucose is simultaneously transported and activated by the PTS system. And the PTS system stands for phosphotransferase system, so PTS, okay? The PTS. PTS is, instead of using ATP to sort of activate glucose, what it's going to do is it's going to use PEP right here. Remember, we talked about in our last lecture, PEP, phosphoenolpyruvate. is a super high energy phosphate donor, okay? And PP will then donate the phosphate to some partners, okay? I don't really care if you know these partners or not. And they're going to do this phosphorylation chain, okay? So it's gonna, this guy phosphorylate that guy, becoming this guy then phosphorylate this guy, becoming that, blah, 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 okay? So on and so forth. And essentially, this sort of phosphorylation potential, so this phosphate potential, is going to be used to drive the uptake of glucose, and then simultaneously you phosphorylate glucose into glucose 6-phosphate. Okay, so it's going to do the transport and the activation in one step. Okay, so this is called the PTS, the phosphotransferase system. Okay, so it's primarily found in bacteria. Now, obviously, you should sort of recognize the fact that PEP has a higher energy than ATP. Okay. Also, what you should notice is here, in this mechanism that I described, converting glucose to glucose 6-phosphate, okay? If you use active transport, you then consume a total of 2 ATP, right? Because if you use active transport, here you use 1 ATP, and here you use 1 ATP, okay? And going from PEP to bring in glucose, okay, we will discuss in maybe next week. Turns out PEP to pyruvate is the same thing as 2 ATP, okay? So this energy is sort of conserved. All right, so second reaction, all right, it's called the phosphoglucose isomerase. Obviously, phosphoglucose, okay? The name comes from glucose 6-phosphate, so now it has a phosphate on glucose, so it's going to be called phosphoglucose, and then isomerase does what? Isomerization, okay? All right, so isomerase, what it's going to do is it's going to turn glucose 6-phosphate into this thing called fructose 6-phosphate, okay? Glucose is what kind of sugar and fructose is what kind of sugar? Right, glucose is what type of sugar? Aldose, okay? Fructose? 不是 aldose, ketose, okay? So the second reaction is phosphoglucose isomerization, or isomerase. RXN stands for reaction, okay? Reaction 2. And essentially, let's start with glucose. All right, this guy is G6P, right? Glucose 6-phosphate. And the first thing that I'm going to do is I'm going to turn this Haworth projection into a Fischer projection. Do you guys remember how to do that? No? Maybe? Maybe okay? Fischer projection. Fischer projection. 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon, and the last one is this 6 carbon. 6 carbon. Carbon 6 is obviously CH2, phosphate. Why? Because it's glucose 6-phosphate. Okay? It's glucose 6-phosphate. Therefore, the 6 carbon here, this guy is C6. So it has a phosphate on it. This is 5, 4, 3, 2, 1. Okay, now, first carbon is a what group? It's a aldols, right? So C1 is going to be a aldehyde. Yeah, maybe? Make sense? Yeah, makes sense, right? All right, so this guy is an aldehyde. Okay, now, this one here is C1, right? Why is that? Because it has 2 bonds, right? So here's one and here's another one. So it has 2 bonds to oxygen. So 2 bonds to oxygen. So this guy is C1, C2, C3, C4, C5, C6. Okay? All right. So my question for you then is C2 is here, right? The OH is going to be on the right side or the left side? It's going to be on the right side because anything that's pointing down is going to be on the right. Okay? Remember that animation I showed you, right? how it go from Fischer projection, it's going to like twist, 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 and then form the Haworth projection? Yeah? Maybe? OH, OH. Hydrogen, hydrogen, hydrogen. Okay, so this is glucose 6-phosphate in its Fischer projection form. Okay, so it's 6 carbons. Now, the first thing that we're going to do, is we're going to have an enzyme. So because we're drawing mechanisms, so I'm just going to simplify this and say enzyme somewhere has a basic residue that has lone pair electrons, okay, or some kind of electrons. This can be, serine, cysteine, whatever, okay, anything that can serve as a nucleophile, okay? And this nucleophile will be used to pull off this proton, okay? What is it so special about this proton that I can pull it off, that I can deprotonate it? 这个很重要，这很重要。这个 proton 啊 ，OK， 为什么我可以拔这个 proton？ 因为不要忘记 C2 这边是一个 carbon 哦。那我们不是说hydrogen bonded to 一个 carbon， 它是基本上你没办法拔下来 ，right？ Because it has a very high pKa， 对，它 pKa 很高。那为什么我这边就画给你看，说我这个 proton 可以被拔下来 ？What is it so special about this particular proton? So this is carbonyl. This is alpha carbon. This will be beta carbon. This will be gamma. Okay. So it's an alpha carbon. It's a hydrogen on the alpha carbon. So if you were to pull this off, okay, pull off this particular proton, the electrons can go to forming a pi bond between the carbonyl carbon and the alpha carbon, okay? And once the electron goes here, then here, the electron here can then go up to oxygen, right? And this is why, our last lecture, I spent so much time talking about oxyanion formation. So I'm going to call this R, okay? I'm just going to abbreviate the lower part. So here you're going to have an hydrogen. Here you have an oxyanion looking thingy. This turns out like an enolate. Here OH, and then here is your, sorry, not enzyme. Here's your R group. Okay, so R is the rest of the molecule, and then obviously you see how I pulled off the proton here. So now this particular carbon lost a proton and now becomes an alkene kind of, and then the double bond that was initially here is now on the oxygen. Okay? Now, like always, you have this oxyanion looking thing. Okay? What are the two things it can do? Grabbing a proton and coming back down, right? Okay? In this case, if it were to come back down, okay, if it were to come back down, you just generate your aldols, right? If it comes back down, if it pops back, then you regenerate the aldehyde, okay? I don't want that. I want to get a ketone out of this reaction, okay? So it's going to take option 2. It's going to grab a proton. Turns out it's going to grab this guy, okay? And then the electrons pop over here, okay? So then what you'll get is, you move the negative charge from the top oxygen to the second oxygen, okay? Then you're going to do the popping back down business, okay? As this particular electron forms a new bond between carbon and oxygen, the extra electron here is going to go somewhere, okay? And then it's going to go to the proton that was pulled off by your enzyme. Okay, so it's going to grab this guy, okay, like that. And then you regenerate your enzyme, okay? And what do you have it? You have on top it's what is this? Here you have a carbonyl. You have a carbonyl, and then the rest are going to be the same. Okay? And the rest will be the same. So you turn an aldehyde to a ketone. Okay? All right. This thing is fructose 6-phosphate. So I just call it F6P. Right? So now, for my purpose, my exam purpose, if you cannot remember the full name of the molecule, okay, at least remember FAOP, okay? Well, F6P. G6P, F6P. By the way, pay attention to this, the delta G value here. Isomerases in general, they have very near equilibrium delta Gs, okay? This is completely reversible, right? Because the delta G, standard delta G is about like 2 kilojoule per mole, so that means it's pretty stable, okay? Pretty reversible. All right. Next reaction, I don't really want to talk about the mechanism, and that is because it's just another kinase. Okay, it's just like the previous one. Reaction 3 is called phosphofructokinase. So clearly you can get it from its name, phosphofructose. So it's a fructose phosphate kinase. So you're going to add another phosphate onto fructose phosphate. So you will generate a product that's called, right here, fructose 1,6-bisphosphate. I don't know why it's called bis, not biphosphate or diphosphate, but it's called bisphosphate. Okay, so it's fructose 1,6-bisphosphate. Okay, this is the what is it? Phosphofructokinase. Okay, and the way it works is very simple. You have F6P. Okay, and on F6P, the carbon 1. Okay, remember for ketoses we name it. We start numbering the carbons based on whichever end that is closest to the carbonyl. Okay, whatever. Okay, so it still has a free OH on carbon 1. Right, so it's going to, do the same business as the previous one. So I'm going to skip a lot of stuff, but hopefully you can understand it. Lone pair, attack phosphate, electron jump up, okay, jump back down. This one leaves, okay? So then you kick out. This is ADP. You kick out ADP. And then you're left with basically F6P, carbon 1, O-phosphate. Okay? And then this is F16-bisphosphate. Okay, so F16BP. Okay, so mechanism's completely identical to the first reaction. Okay, so you have a nucleophilic attack onto the terminal phosphate on ATP, and then the electron go up on the oxygen, comes back down, and then the bond between the terminal phosphate with the second phosphate on ATP will break. Okay, so then you release ADP, and then you're left with a phosphorylated sugar. Okay, in this case, now it has 2 phosphates. Okay, this reaction is very important. Okay, this reaction is actually very important. This is what we call the committed step. Okay, so the committed step of glycolysis. What it means is after fructose 6-phosphate is phosphorylated into F16BP, this molecule is signaled for degradation to pyruvate. Okay, so it's not going to go anywhere else, typically. Okay, it basically tells the organism that, hey, this sugar, I'm going to degrade it. Okay, right? Because why not the first reaction? Because F6P, we can use it for other pathways and for other things. But here, fructose 1,6-bisphosphate, it's going to be used for glycolysis. Okay. Of course, some intermediates in glycolysis can also be used for other biosynthetic purposes. But essentially, this is like the first, not first, but the committed step for being broken down. Okay. So after this particular step, your 6-carbon molecule will be degraded into 2 3-carbon molecule. If we were to look at the overall reaction here, okay, I know it's a little small, but if you take a look, we went from glucose, okay, 6-carbon aldols, first activated into glucose 6-phosphate, and then we did an isomerization, and then it becomes fructose 6-phosphate, and then we did another activation into fructose 1,6-bisphosphate, and essentially what you should notice is that now the molecule looks somewhat even, okay, like that. Okay, now you have sort of 3 carbons on one side with a phosphate, 3 carbon on one side with a phosphate. Okay, so that's sort of the goal, right? We want to prep the molecule and get it ready for aldolase. This is actually very hard to guess, okay? Obviously, ACE means that's an enzyme. Aldol, where have you seen aldol? It's this thing called, Aldol condensation, right? Claisen condensation, yeah, maybe, okay. Remember in our last lecture, we talked about this aldol condensation, which can turn, you can react a carbonyl with an aldehyde, right? This guy's an aldehyde. This guy is a carbonyl, right? And you can form a bond between the alpha carbon on the carbonyl with the carbonyl carbon on the aldehyde, right? So you're going to form a bond between here and here, okay? So it's called aldolase because it catalyzes a reversible reaction, okay? A reversible aldol reaction, okay? That's why it's called aldolase, right? So if you take a look at the slide here, what you should notice is that this is sort of the first reaction where we break the 6-carbon molecule into 2 3-carbon molecule. And at the end of the reaction, you will generate 2 sugar phosphates, okay, 2 triose phosphates. Remember, triose, glycerolaldehyde 3-phosphate is considered the simplest sugar that you have. I'm going to talk a little bit about naming, okay? Dihydroxyacetone phosphate, okay? DHAP. All right, acetone, you know the structure? This guy is acetone. Acetone, okay? Dihydroxyacetone is acetone with dihydroxy, right? So you have dihydroxy, okay? Acetone and dihydroxyacetone phosphate. Here's an OHO phosphate. Okay, so that's it. So the way these names, you should be able to guess them. Acetone, dihydroxyacetone, dihydroxyacetone phosphate. Now, glycerolaldehyde 3-phosphate sucks for you. You just have to memorize it. Can't help you there. But it's actually related to glycerol, which is 3-carbon alcohol. But here it's an aldehyde. Okay.